without further ado, I'm going to let Alicia take it away for our presentation today.
Awesome. Thanks so much. Let me get my slides up here. And thank you everybody for joining us live today. I know that you're all very busy practitioners. And so, the hope today is that we can really hopefully review some foundational concepts, bring in some things that maybe you've learned in the past, but maybe have forgotten, but are still valuable and important to understand when you're working with clients, especially in the realm of GI disorders. So this is our agenda for today. The first half of the webinar is really going to be kind of an anatomy and physiology review. Some of you might feel like you've gone back to your university training in anatomy and physiology, which may or may not be a good thing for you. But we're going to go back through the structure and the function of the intestines. We're going to talk about and review the different cells within our GI tract and what they do and how they interact with each other, especially like the immune system. We're going to talk a little bit about the GALT. And then we're going to go just real briefly through some basic anatomy and physiology of the gut-brain axis. So that'll be the first part, is going to be more anatomy, physiology. And then the second half of the webinar is going to focus on walking through the digestive organs and the digestive process from top to toe. So we're going to talk about the brain and the cephalic phase of digestion all the way down through to the colon. And the reason for that is because as part of this foundational series that Angelica was mentioning that we're doing once a month, these webinars will build upon each other. So, this one we'll have today, there'll be one in November, one in December, and one in January. And so whenever you see a slide that has a blue star, that's a concept that I'm going to basically kind of introduce and plant some seeds, lay the foundation, but those concepts will be further expanded upon in later webinars. So just pay attention to some of those. I might not go real deep into some of those slides or some of those concepts because, like I said, they will be mentioned later on in future webinars. So think of this as a 4-part series, like a course, to where at the end of that course, you're going to walk away with some really good tools and hopefully a good refresher and an understanding, or just a review or re-understanding of what you might already know, but maybe in a little bit different light. So let's go into intestinal anatomy and physiology. So we're going to talk about just some real basic stuff here, just real basic. So the human body, we have different levels of organization. So we start out with cells. Cells come together to form tissues. Tissues come together to form organs. And then organs come together to form organ systems. And so the human organ systems have different biological processes and functions. So they have, they produce biological materials. They might facilitate the elimination of waste as the digestive system does, or they might help to maintain body temperature, pH, or other conditions. And the GI or the digestive system has 3 main functions: digest food, absorb nutrients from that food, and eliminate solid waste. Now, we're going to go into a lot more about like the immune system and the different functions of the microbiome and whatnot. A little bit in this webinar and then definitely for the next one. But in terms of the digestive system, 3 main functions, it's primarily that. And then these organs of the digestive system are really broken into our main core. So the esophagus, the stomach, the small intestine, and the large intestine. But then we have our accessory organs like the pancreas and the gallbladder and the liver. And so we're going to go through some of those and just review how they work together in synergy with each other so that we have this really lovely symphony of the digestive system working together. So let's go through the importance of an epithelium. So an epithelium is basically a sheet of cells that covers either the body surface, like we think about the skin epithelium, or it lines a body cavity. So this can be the intestinal epithelium, this can be the lung epithelium, this can be the stomach epithelium. So basically is lining that surface, that cavity or that surface. And it also forms a boundary between environments. And so an epithelium, what it's doing is it's either protecting, it's helping with absorption, it's filtering. So think about like kidney epithelium, excretion, secretion, and then sensory reception. So there's functions of what an epithelium is supposed to do with whatever organ system it's covering. Now, all epithelium are bound together by tight junctions. So we talk about tight junctions as it relates to the intestine, but different epithelium, like in the lungs, have tight junctions as well. So one of the things that's talked about some in the literature and in some of what we discuss is that if you have leaky gut, if you have leaky epithelium layers in your gut, You could have leaky brain, you could have leaky lungs, you could have other areas of the epithelium throughout the body that can also be compromised to a degree, because different factors that compromise tight junctions in the gut could also compromise the tight junctions in other areas of the body. And so therefore, you can have other epithelial layers that are breaking down. And an important concept to understand when it comes to the lumen, which is that innermost part of the intestine, is that as long as food and proteins and different microbial byproducts like LPSs or inflammatory cytokines, as long as those are contained within the lumen, then it's technically still outside the body. Nothing really goes into our body until it has crossed through that epithelial barrier. And that is why we have a semi-permeable membrane, because we want certain things like nutrients and certain anti-inflammatory cytokines to get through, but we don't want for large food proteins or microbes or LPSs to pass through. So as long as it's in that epithelium, it's technically outside the body. And it's that lumen that's really colonized by that mass amount of microbes when we talk about the intestines. And you're going to go more into the microbial balance in the microbiome in the next webinar. So we're just going to touch on it today. So let's go through this particular schematic here. So just kind of thinking higher level of the intestinal tract, and we're talking about mostly the small intestine here. So there's 4 layers. You have the mucosa, which is where we have the epithelial, here we go. So you have the mucosa, which is where we have those epithelial cells, which is where, they're secreting some mucus, but, they do other things, and we'll talk about that here in a second. But the main function, again, of the epithelium and the mucosa is to absorb and transport those nutrients and to help to keep those tissues nice and moist and to protect the body from pathogens. We then have a submucosa, which is a thinner, kind of more of a collagen rich matrix, and it's supporting the mucosa. You then have a muscular layer, which is more with like peristalsis and the migrating motor complex. So it deals more with the motility aspect of the intestines. And then you have the adventitia, which is a smooth muscle or smooth tissue membrane that has 2 layers, and it helps with secreting certain fluids. So just high level, there's 4-ish layers of our intestinal, our intestinal tract. And then if we think about breaking it down a little bit further, we have this epithelium, which is that single cell layer. So just one cell separates the contents of the lumen from the rest of the digestive system. And we're going to talk about some immune cells and why we really want to ensure that we have an intact epithelial layer, because that's our only barrier that's keeping things out that should stay out and helping to bring things in that should come in. Now, enterocytes are our small intestinal cells or intestinal cells. They help with nutrient absorption. They help with secreting certain immunoglobulins like secretory IgA. They have the villi there that are little fingerling projections that help with that absorption. You then have Paneth cells, which these are only in the small intestine. So they help to make our own natural endogenous antimicrobial proteins. So we have a system in our GI tract, in our epithelial layer, that is supposed to make our own defense mechanisms. Beta defensins is an example that help to deal with any sort of pathogens that might be coming in. We're going to talk about, next to the Paneth cells here, you can see that there's a Peyer's patch, which is also primarily in the small intestine. And we're going to break down the Peyer's patch a little bit more, because that plays a huge role in immune balance in the small intestine. We have goblet cells, which help to secrete mucus. We have some goblet cells in the small intestine. We have a very large amount of goblet cells in the colon, because there's some differences in the mucus barriers between the small intestine and the large intestine, and we're going to talk about those differences today. And then we have neuroendocrine cells, and this is where these are the cells that help to activate certain nervous system responses, and they help to initiate certain digestive actions and protective responses. So these are some of the cells that are within that epithelial layer of our intestine. Now, there's some other cell types. We have exocrine cells. Now, these are just cells that are secreting things. So they secrete mucus like goblet cells, or they might secrete things like digestive enzymes in the small intestine. And then we have endocrine cells. And these are cells that are secreting certain hormones like cholecystokinin or CCK and secretin. We're going to talk about these two hormones when we get to reviewing the digestive system, because our GI tract secretes like 16 different hormones that help to regulate and signal various digestive processes. So our intestinal cells are basically part of that process to help to regulate our digestive system. Now, these secretions are regulated by chyme. So the more chyme we have, the more secretions. And just as a review of what chyme is, it's just basically this lovely mass of undigested food and different, stomach acid and gastric juice and pancreatic juice. So it's this lovely mixture that is formed in our stomach, and then it gets dumped into the small intestine. And when it gets dumped into the small intestine, it actually triggers a series of signals that then leads to further digestive processes as that food moves through the intestinal tract. And we'll talk about that when we get to the second half of the webinar. But I wanted to put it here because it ties in with those cell types that we're discussing in the small intestine. So again, this is a high level view of going back to the epithelial layers. So we have an epithelial layer in our stomach, and then we have an epithelial layer in our small intestine and then our colon. And I want to point out, if we look at the differences here between the stomach, the small intestine, and the colon, we can see that the stomach, you've got this kind of nice, it's sort of a medium layer. It's not as thin as what we see in the small intestine, but it's not as thick as what we see in the colon. But it's definitely more of a robust layer because we have such an acidic, that gastric juice is so acidic. We want to have a nice mucus layer to protect that epithelial layer in the stomach. The other thing that's different with the small intestine here is that, that thin layer of mucus, but again, the Peyer's patches. This is important. We're going to talk about how the immune system differs in the small intestine versus in the colon, because the small intestine has a different role to play when it comes to distinguishing, food and proteins and bacteria that we're swallowing that are coming in from the outside. We have to have a really robust immune system in our small intestine to deal with some of those external things that are coming inside the body. There's also the Paneth cells. So remember, those are the cells that create those antimicrobial peptides. Those are unique to the small intestine. And then these M cells, which play a role with the Peyer's patches. They play a role with antigen detection and with pathogen detection. And those are also in the small intestine. So, I wanted to go through and discuss the differences of the intestines as it relates to the small intestine versus the colon, because sometimes I think we sort of lump it all together. And there's actually, differences that are important to understand so that as we discuss interventions later on, and as we talk about various strategies, you can understand why certain things might be happening in what area, and then why people might be struggling with certain symptoms, depending on the site of where they're having their imbalance within their gut. So again, here's the mucus layer. We have two mucus layers within the colon. So if we go to the colon, we can look at this a little bit deeper as it relates to the mucus barriers and as it relates, I'm going to move my little video here, as it relates to the functions and it relates to the microbiome. So again, as a review, this mucus layer is pretty thin here in the small intestine, and it's not attached to the epithelium. Whereas the colon, we have an inner and an outer layer. So the inner mucus layer is free from bacteria. It is the only part of the body that is completely sterile. We used to think that things like blood and urine and the vaginal tract and the uterus and various other parts of the body, we used to think that those were sterile. Actually, there is some microbes in the blood. There's microbes in the urine. There's some microbes in the vaginal tract, as we surely know now, and in the uterus. But this is the one area that is completely sterile. And there's a reason for that, because when you think about the massive amount of microbes and microbial byproducts that are in the colon, it's the mucus layer that's the only thing that's going to separate all of those microbes, all of those foreign proteins and whatnot, from getting to where we have the largest concentration of our immune cells. So we have the largest concentration of microbes and the largest concentration of our immune cells in the intestines. So we have to have a nice, robust mucus layer to separate those to some degree so that there's some regulation at what's really getting through and triggering an immune response. And then the outer mucus layer of the colon does have some microbes. This is where kind of, a lot of those microbes live, but But it's a really nice thick layer that we have going on in the colon here. So again, here's that inner and that outer mucus layer in the colon, very different from what's happening in the small intestine, which makes sense because in the small intestine, one of its main roles is to digest and absorb nutrients. So we don't want to have a really strong, thick mucus layer in the small intestine because that's going to interfere with the function of the small intestine. But we want to have some mucus layer in the small intestine because if we don't, then you can have more antigen interaction with various immune cells, and that can overstimulate the immune response and cause a loss of what's called oral tolerance to things like foods and whatnot. So it's all about balance and about the function of the various parts of the body. So here's just a recap, just a little chart that I made for you that you can quickly reference back of just what some of the differences are between the large and small intestine. So again, the function of the small intestine is that nutrient absorption, immune balance, and then the large intestine is mostly absorbing water and electrolytes, and it's also a site where we have vitamins that are made by our bacteria. The mucus layer is thin and kind of a single layer with some bacteria in the small intestine versus being thick in the large. There are some goblet cells in the small intestine for that kind of thinner mucus layer, whereas there's a lot of goblet cells in the large intestine, so you can get that thicker mucus layer. And there's more immune cells in the small intestine. There is immune cells in the large intestine, but just not quite to the degree of what's in the small intestine. But also the anatomy is different. So we have the villi, or those fingerling projections, and shallower crypts. Whereas in the large intestine, we don't have any villi and we have those deeper crypts. We have more of which you can see here, villi being here, crypts being here. So again, there's a difference in how the anatomy actually looks as well. So let's go through and discuss the immune system and immune system regulation. So I think this is it's a really interesting article anyway about just IBS and the relationship between the immune response and IBS, and how we need to think about it being part of the small intestine and not just isolated to the colon when it comes to pathology of IBS. But if we think about just, again, the function of these two systems, the colon, the immune system is more focused on dealing with the massive amount of commensal bacteria that we have. Trillions of bacteria are in the colon. And so there has to be anatomy and physiology that can deal with that. Now, the small intestine, the immune system is more focused on discriminating, okay, is this a friend? Is this a fro? Is this a chicken protein? That's okay. Is it a Campylobacter bacteria? That's not okay. And so it's trying to constantly discriminate between what do I want to digest and absorb and what do I want to have the immune system try to deal with. So if we look at this chart over here, we can see that the different, like the villi length, the goblet cells, but definitely certain things like secretory IgA levels, much, much higher in the further upper part of the small intestine, and it kind of decreases as you get to the colon. T helper 17 cells, which as long as these are in balance, it can lead to things like homeostasis. It's when there's imbalance of the immune system that it can lead to things like autoimmunity. But you have a lot more of that concentrated in the small intestine versus as in the colon. And then these antimicrobial peptides. So again, remember those Paneth cells are located only in the small intestine. So you're going to have a lot more of those natural antimicrobial peptides in the small intestine versus in the colon, because in the colon, you have a lot of microbes. You don't need to have a ton of antimicrobial peptides there necessarily. That could be interfering with some of the function. So I think it's interesting to see here, and again, goblet cells, you see how that concentration greatly increases in the colon. So it's interesting how we have kind of this dynamic, these opposite ends of the spectrum, so to speak, but yet they work together in unity. And if one area is in balance, it can lead to imbalances in other areas as well. All right, so on that note, let's talk a little bit about the GALT, or that gut-associated lymphoid tissue. So 80% of our immune system is in the gut, and we have a ton of different immune compartments and immune cells in the GI tract. And so when we think about the immune system, the GALT is just part of the totality of what the immune compartments contain. So we have these certain things called mesenteric lymph nodes, which we'll talk about. Then we have the GALT, which includes Peyer's patches in the small intestine, and then these isolated lymphoid follicles, which are both in the lower part of the small intestine and then throughout the large intestine. We then have the lamina propria, which is the area just right underneath that intestinal epithelial layer, just right underneath that. That's where a lot of our immune cells are. And then, of course, we have the epithelium, because the epithelium is that first barrier that anything within the lumen is going to have to breach first to get down into these lower parts of where the immune system is. So it's really all of these things working together that then comprise the immune system within our gut. So let's talk about Peyer's patches. This is high level view, just kind of an overview. So they are the immune cells in the lamina propria. So again, the lamina propria is this little bit here. It's right underneath this epithelial layer, so that single cell layer I was talking about. And the density increases as we go towards that terminal ileum. It identifies antigens and it produces antibodies. And it increases to a maximum between the ages of 15 to 25. And then after the age of 25, it starts to decrease. So we start to decrease in the number of Peyer's patches that are located in our intestine as we get older. And it consists of things like B cells, T cells, plasma cells, mast cells. A lot of other immune cells are part of these Peyer's patches in our small intestine. So if we break down the anatomy, we'll go through the anatomy first, and then we'll go through the function next. So this follicle-associated epithelium, or this FAE, basically, this is just a certain section of the intestinal epithelium that's just special. It's special because it overlays the Peyer's patch, and it's sparsely covered by mucus. And again, because of the way the immune system has to work to be able to deal with any potential antigen or pathogen, you want to have that kind of sparse covering of mucus, not non-existent, because you want to have some degree of separation, but you don't want it to be, super thick. And so you then have these M cells, which basically form pockets, and that allows for various antigens to come into contact with B and T cells, and then also it helps with free and bound IgA antigens into the Peyer's patch to then deal with it. You then have, those are the B and T cells, and then you have this SED, or the subepithelial dome, which is where you have your dendritic cells. So dendritic cells are our antigen presenting cells. So when something comes in, the dendritic cells are there constantly sampling to say, okay, friend, foe, and then they go and they present it. And then that's when we have like the various immune responses that happen to either deal with that antigen or to say, nope, we're good, you're fine, it's just a chicken protein. So that's the anatomy. You have this special epithelium, you have these M cells, which are pockets that then regulate the response of the B and T cells and secretory IgA production, because remember, we saw how IgA is much higher in the duodenum, the jejunum, and the ileum than it is in the colon. And that's partially because you have these Peyer's patches. So the function of the Peyer's patches here is immune surveillance. So the process of what's happening is you eat a bad piece of chicken, you have some Campylobacter, or you have you ingest something that really shouldn't have been ingested from food. That pathogen meets an M cell, and that M cell then kind of takes it down. It gets presented to a dendritic cell. That dendritic cell then presents it. The lymphocytes go, oh my gosh, it activates the immune response. And then what happens is you have these mesenteric lymph nodes that then amplifies that response. Now the B cells, they then differentiate into IgA, which then can go and bind to that pathogen and can deal with it in the lumen to bind it, to inactivate it. And then the bottom line really when it comes to these Peyer's patches, you walk away with nothing else from this, is that they help to keep our intestinal bacteria at appropriate levels. They help to deal with pathogens and to keep them, at bay. And they also help to prevent infection. So this is why our small intestine has much more of a robust immune system than what's found in the colon, because of what it's having to do to differentiate what's coming in from the outside. So whatever we're breathing in, swallowing, ingesting, that's what these Peyer's patches are really dealing with. Now, when we get down to that, more distal part of the small intestine, which is the ileum, and into the large intestine, that's when you start to have a different set of immune, a different kind of galt that starts to show up, and that's this isolated lymphoid follicle. There's technically two different types. Don't get bogged down into the details, but essentially you can see here in the picture that you have mucosal and submucosal isolated lymphoid follicles. So you have some mucosal and the Peyer's patches kind of in that terminal ileum. And then once you get to the ascending, transverse, and descending colon, that's when you get to the submucosal ILF, and then you have more of the mucosal and some of the submucosal ILF as you get down to the sigmoid and lower part of the colon. So you kind of switch the immune type, the immune GALT type when you get to the colon, but look at how much smaller it is. It's much, much less in terms of like cells or fewer dendritic cells, because once all of those food proteins and whatever's been digested and ingested, by the time it gets to the colon, most of it should have been dealt with. So, there's not quite the level of antigen sampling needed when you get down to the colon, versus you can see with the Peyer's patch, this is a much greater area with much more complexity and number of these various cells. So just wanted to point out some of the differences there between what's happening in the small intestine versus the large intestine. There's still definitely immune surveillance in the large intestine, and it's still absolutely, very important. It's just the number is a little bit smaller, or the size is a little bit smaller. Okay, so why am I spending all this time on this? Well, because of intestinal permeability. And I think probably most of us here know or have heard about intestinal permeability or leaky gut. But I want to just review it quickly and briefly. Again, this is going to be, you see this as a star. This is a starred slide. So this will be a concept that you're going to hear again in later webinars. But again, as I mentioned, all epithelial cells, all epithelial membranes have tight junctions. And when we talk about the gut, we talk about these tight junctions that help to reduce hyperpermeability. You have some semi-permeability because you want to have things pass through freely, in and out, the things that are supposed to pass through in and out. But you want to have some regulation so that you're not just allowing anything and everything from the lumen to come through and activate the immune system. So you have these tight junctions, you have occludin and claudins, which are these kind of between cell, these cell-cell connections that help to connect these cells together. And then you have the zonula occludins or zonulin, which is basically the intracellular proteins. You then have the mucus barrier. So this is the second part. So you have tight junctions that are like shoelaces binding the cells together. And then you have the mucosal barrier, which we've talked about with the mucin from the goblet cells. You have antimicrobial peptides. Remember those PANF cells I was talking about? And then you have IgA, and basically this barrier or this mucosal barrier is acting as that no-fly zone, or that little barrier between the lumen and the intestinal epithelium, that single cell layer. So if you have any disruption in the tight junctions or in the mucosal barrier, that's when you get that intestinal permeability. It's not just necessarily one or the other. Typically, it's going to be both, because if you have certain conditions that are breaking down the tight junctions, you're probably going to have those same conditions that are going to be degrading that mucus barrier. So they typically will go hand in hand, but they both will lead to that intestinal permeability. And that's when you get that translocation of microbes. You get the release of LPSs that can get out into circulation and can cause immune imbalances, can cause systemic inflammation, and that's when things start to break down. The other important piece of this is, probably most of us on this call deal with clients who have autoimmunity. And in autoimmunity, you have to have three things present. You have to have the genetic predisposition. You have to have an environmental factor. So that might be a poor diet, that might be medications, that might be a viral pathogen or some sort of heavy metal toxin or something that is the trigger. And usually, and especially when it comes to like diet and medications, that can lead to dysbiosis, which can then lead to the epithelial breakdown, and then that can lead to more of a pro-inflammatory response. So when you have these genetic predispositions that's then triggered or perpetuated by an environmental factor, that then starts to lead to that dysbiosis and some of these microbial imbalances that eventually can lead to leaky gut syndrome. And leaky gut syndrome then enhances that bacterial translocation and enhances the progression of LPSs into circulation. And it's leaky gut that is in that third pillar behind genetic factors, environmental factors, and then you have to have that leaky gut that then basically causes an autoimmune condition that may have been dormant, or maybe someone just had the predisposition for it, but it's that leaky gut that then triggers it to actually manifest. And that's when we get that immune imbalance, and we get things like celiac and various other Hashimoto's and IBD. But also, too, we know that leaky gut underlies many, many other things that are not necessarily autoimmune in nature. So metabolic disorders, mood disorders, GI symptoms, skin conditions, chronic migraines, brain fog. Those are all things that can also have leaky gut as their base because we have such an immune presence within our GI tract. So let's talk a bit about the gut-brain axis and the vagus nerve. So the vagus nerve is this nerve that connects our brain to our gut. And when we talk about the gut-brain axis, we're talking about this umbrella term that's really a bidirectional communication, but it includes the vagus nerve along with other things like the endocrine system and the immune system, and of course our microbiome. So the gut-brain axis is really this whole collective that have to work together to then allow proper signaling of the gut and the brain to each other. The enteric nervous system is fascinating because we have more nerves in our gut than we have in our spine, and it produces more than 30 neurotransmitters. So we usually think about neurotransmitters in the brain, but we're definitely producing a lot of neurotransmitters and also neurotransmitter precursors in our gut from our microbes. There's also hormones and certain peptides that are released by the enteric nervous system that can then get through into circulation and cross the blood-brain barrier and can interact with the vagus nerve and lead to various signaling. But also, this is super interesting that about 90% of the signals between the gut and the brain are actually coming from the gut. So those are what are called afferent, which is basically gut to brain signals. It's about 10% that are coming from brain to gut. So we have a whole lot more communication that's coming from the down up versus the top down. So again, it just speaks to that importance of, how much our gut and what's happening in our gut with the various signals are influencing how we're thinking, how we're feeling. And again, this is a starred slide. So you're going to go more into gut-brain axis communication and more gut-brain axis information in future webinars, but you can have this as a reference to refer back to. So the vagus nerve innervates the pharynx and larynx, so it deals with swallowing. It also helps with reducing heart rate. This is why breath work, if people are stuck in a sympathetic state, this is why breath work is so important, because we can help to shift people into a parasympathetic state, help to calm that heart rate. It helps with the intestines in terms of regulating the contraction of smooth muscles and glandular secretions. Again, those vagal afferents coming from the receptors in the esophagus and the stomach and the various GI organs up back to the brain. And it's responsible for things like digestion, heart rate, respiratory rate, and then also vasomotor activity, so swallowing, vomiting, or sneezing. So this is why, again, there's a star here, so we'll come back to it. But like if you think about vagal nerve exercises, typically the things that are recommended are things like gagging, gargling, singing really loudly. And if you have people who can't gargle or they have an overactive gag reflex, then that's a signal that there's something that's happening with the vagus nerve that is not firing properly. So the vagal nerve exercises will be even more important for them if they are finding that, oh my gosh, I can't even gargle. They're not getting the signals between the brain and the gut that are necessary to basically dictate the way the digestive system is supposed to work. All right, so let's see how we're doing on time here. We're now going to switch gears. I just want to do a quick check-in with how everybody's feeling. If you guys feel like you've just gone through your anatomy and physiology course, we can talk through that. But just think about some of these various things that we've discussed and how it might be relating to your patients. We're going to talk more even about that now. When we talk about digestion, I'm going to bring in some clinical pearls for you as well. Okay, so the brain is important to digestion, not because of the gut-brain axis only, but because of this thing called the cephalic phase of digestion. So the cephalic phase of digestion, I think we sometimes forget about. But if we look at some clinical pearls, 20% of our gastric secretions happen before we've even eaten a meal. It's mediated entirely by the vagus nerve. So when we see food, smell food, think about food, and start that very first beginning of tasting food, that vagus nerve is already starting to act on the stomach by releasing acetylcholine. That then tells the parietal cells, hey, you need to start secreting some stomach acid because we've got some food that's about to come in. And so think about your people who are in a sympathetic state all the time. They are not in a rest and digest state. And what's happening with that signal of the vagus nerve to be able to tell the stomach to start releasing. And if you're thinking and relying on 20% of your gastric secretions are supposed to happen before you've even eaten, then what are some things we need to think about when it comes to our patients and our clients? So what to look for in a client history? Are they eating mindlessly? So are they not really getting into that cephalic phase of digestion? Are they drinking their meals? So are they just throwing a smoothie together, or just grabbing a protein shake from the fridge, or just trying to drink a liquid meal because they're fast and stressed and they have no time to sit down and eat? Then all of a sudden you have this whole, a bunch of nutrients coming into the GI tract, and the stomach's like, well, wait a minute, we didn't get that signal. We had no idea that this was even coming. So think about, if someone loves their smoothie in the morning, make sure that they're going through a whole process of chopping the vegetables, chopping the fruit, making sure that they're taking in, sensory, feeling the strawberries, smelling the blueberries, making sure that they're going through some process so that they can start to activate. If they're not going to be cooking something, that they're still going through that process to activate their sensory inputs. Are they stressed at meal times? Have they had a head injury? Have they had a head injury that's affected their vagal nerve tone? Do they have poor appetite, or no interest in food or food prep? So they're not really interested, and they're not getting that initial activation. Do they have deficiencies in taste or smell? So do they have Parkinson's or really bad allergies? So they're not even able to smell the food or smell or really even taste, because our smell and our taste are intimately connected. So these would be some questions to think about when you're thinking about assessing their cephalic phase of digestion. All right, now with the mouth and the oral cavity. So chewing is super important. I'm sure we all could be better with our chewing. I know I could be, but we need to chew really well. So each bite about 30 to 40 times to properly break down food before swallowing. So if we're not chewing enough, we're going to probably have more bloating, gas, diarrhea, GERD, cramps, and nausea. So those are just some things that can be a very low level hanging fruit for some of your clients, that if they're having a lot of these struggles, the first thing you can do is look at how much they're chewing. The other thing when it comes to the mouth is the saliva and the person's saliva production, because saliva plays a big role in that initial phase of digestion. So with amylase, it's breaking down starches, which is actually it's identical to pancreatic amylase. We have lingual lipase, and then we have lysozyme, which is what helps to regulate and keep our oral microbiome in check and not have that overgrowth. But it also helps with the oral hygiene. So, it helps to float away debris. This is also why when you wake up in the morning, you might have dragon breath, bad breath in the morning, because your saliva production decreases overnight, and that allows bacteria to build up, and these various byproducts from bacteria to cause that bad breath. This is why good oral hygiene is really important. But then also the oral microbiome after the gut is our second largest microbial community. So if you think about, we have the second largest here at that first opening of that first site of what's happening. And then we have our second largest in our gut. So there's a lot of microbes involved in regulating the outside world coming in for our protection. But if we have oral dysbiosis, or if we have an overgrowth of certain oral bacteria, then that can lead to various pathogenic and chronic diseases. And here's a list of what those could be. So these are some of the different oral microbes that are associated with certain systemic disorders like diabetes and cystic fibrosis and arthritis. So if you are running BiomeFX tests, if that is a stool test that you have started running in your practice, pay attention to Prevotella. Prevotella is one of the genus that is on that dysbiosis ratio. If someone's really heavily favoring Prevotella, be thinking about their oral microbiome, be thinking about their mouth-gut axis. But also there's many other species that if they're showing up in the stool, they're probably translocating from the oral cavity and getting down into the gut. And that's not a good thing. That means that there's some breakdown and some barriers. There's potentially some dysbiosis. And so you want to be thinking about that. So what to look for in a client history. So this would be things like TMJ. They're probably not able to chew very well if they have TMJ potentially. If they have poor oral hygiene, there's certain medications that can cause dry mouth. So they have that lowering of that saliva. If they have things like Sjogren's or lupus, that also is going to lower their saliva production. Diabetes, they can have more of a predisposition for oral candidiasis because of the hyperglycemia. And then are they really slow in the last to finish a meal? Because if they are, that might mean that they have low saliva production. Now with the esophagus, again, the vagus nerve is going to mediate that parasympathetic activation. We have the sphincter that connects the esophagus to the stomach. And if it's relaxed, then that's when we have gastric reflux. If it's really contracted and tight, that's when we can have dysphagia. And so those are the various things. I don't know that this is going to be new for anybody, but things like alcohol, tomatoes that can relax that LES and can really worsen things like GERD. And then if you think about, there's any signs of impaired, relaxation or increased tone, things like chronic coughing, burping, difficulty swallowing solids, that could cause that. And the other thing that can cause it is mast cell. So if people are having a lot of upper gut, refluxy issues, the excess relaxation or impairment of relaxation can be caused by an overabundance of histamine and mast cell activation. So that's kind of like the top part of our digestion. Now, as we start to go into our stomach, pancreas, liver, and some of the accessory organs, let's just remind ourselves of the various digestive factors that keep microbes in check. So we have our gastric acid, which is really acidic to help to neutralize any microbes. We have pancreatic enzymes, which also have antimicrobial activity. Bile acids, which do the same, small bowel motility, our ileocecal valve, and then the immune system. So these are all the checkpoints that make sure that the types and also the abundance of microbes in our small intestine and large intestine are kept in balance. So if we see imbalance of the large intestine and the small intestine, that probably means then there's dysfunction in some of these upper factors, these upper checkpoints. So let's go through the stomach. So again, there's an epithelial lining, just right there where the mucus, and we really only have mucus cells. We have a lot of mucus cells here, like I said, but we also have gastric pits, and these gastric pits basically lead to gastric glands. And these gastric glands are where we get that gastric juice. And the parietal cells, which is just kind of this little outset here. Parietal cells are what are going to make that stomach acid, and also intrinsic factor, which we have to have intrinsic factor to bind to and absorb B12. We also have chief cells, which secrete inactive, so pepsinogen, which is inactive, and that has to be then activated by stomach acid to then convert to pepsin, which is what we need to break down proteins. So you think about people who are on PPIs, or they have H. pylori, or they have, there's certain patterns that we can see in stool tests of various microbes that are elevated that might indicate, and also certain functions like certain proteolytic or protein fermentation byproducts that might indicate that there's an imbalance of the stomach acid production that's then leading to imbalances further down. And then we have these enteroendocrine cells that secrete various hormones into the interstitial fluid like gastrin. Now, the gastric phase of digestion is about 3 to 4 hours, and it's activated by food coming into the stomach, which again, if you remember from the cephalic phase, that acetylcholine gets triggered further, and that further helps to release that gastric juice. And it's that gastric juice that then starts to ensure that we get that protein activation to then deal with these proteins that are coming in from the meal. And then we also have to have the gastric juice to activate the pepsinogen to pepsin. So this is what's happening when we first start to eat a meal and it starts to go through that gastric phase. So what would be some signs of low stomach acid? If they're burping after eating, if food is sitting in the stomach, undigested food in the stool, constipation, but also too, if they have a lot of ulcers, or if they have a lot of stomach inflammation, if they have any erosion of that mucus barrier that's maybe starting to happen and to cause ulcerations. And that's going to be, relief from carbonated drinks, feeling better when they've eaten a meal versus when their stomach is empty. If they're feeling full after small amounts. So this is just a list. You don't feel like you have to memorize these, but just so you have them as a reference so that you can refer back to them if you have clients that are struggling with some of these. Now, in the small intestine, this is where we have mostly that chemical digestion. And this is, again, that chyme, which is that lovely mixture of digestive juices and undigested food coming into the duodenum from the stomach, and it's starting to mix with secretions from the pancreas and the duodenum. Now, it takes about 5 hours for one meal to be processed through the entire length of the small intestine. So there's also coordination that has to happen between the gallbladder and the pancreas to really get to that full digestive potential. And we're going to talk about some of the signals that have to happen in order for you to get that full digestion in the small intestine. So in that first upper part, which is the duodenum, it's that first part of the small intestine. This is where you have mostly digestive juices from the liver, the pancreas, and the gallbladder. So things like bile and pancreatic enzymes. It's a much more acidic environment because you're getting that chyme that's coming from the stomach. And it's also where you're going to get a lot of mineral absorption, fats to some degree from the pancreatic lipase, B1, B2, and B6. And the jejunum, which is that second part, that's when you're starting to get some more mechanical digestion. You're going to have some folate absorption. And then the ileum, food is going to spend most of its time in the ileum. And you have the ileocecal valve, which is the valve that separates the ileum from the colon. And it prevents that backflow of the intestinal contents from the colon into the small intestine. But this is also where we have a lot of hormone and nerve signals. And this is also where we have primarily B12 and bile salt absorption is in the ileum. Now in the pancreas, this is one of our accessory organs, and the pancreas really has two different functions. We have endocrine function and we have exocrine function. So endocrine being things like glucagon and insulin, but most of it is going to be digestive output. So that's about 80% of the pancreas's job is to help with digestion. And the enzymes, whereas the enzymes in the stomach function at a really nice acidic pH, the enzymes produced by the pancreas into the small intestine, they function at a more basic pH. So they have to have a little bit more of an alkaline environment due to bicarbonate release. So when food's going from the stomach into the duodenum, you get this activation of CCK, which is secreted in the duodenum, and it's enhanced by secretin. Secretin helps to stimulate that bicarbonate from the pancreas to start to help to neutralize that real acidic chyme. And then CCK also helps to secrete or release the secretion of bile. So it stimulates the gallbladder to contract into the intestine. And then we also have the various pancreatic enzymes. So we have amylase, lipase, and then the various protein like trypsin, chymotrypsin, and elastase. So elastase is going to be, you may have seen that on some of the stool tests. It's one of the measures of pancreatic output. So what to look for in a client's history? It would be things like, again, difficulty digesting carbohydrates and fibers, having a lot of greasy, poorly formed floating stool. That can come from either low lipase or it can come from poor bile flow. Poor appetite, if they have a history of diabetes, which is going to put a lot of pressure on the pancreas to be able to excrete the enzymes. Again, low stomach acid. So if you think about if someone doesn't have proper pH in the stomach, then they're not going to get that signal of dumping that chyme, that proper pH of the chyme to dump into the small intestine to then signal the various, secretin and CCK to then tell them to neutralize. So if you don't have that proper pH, enzymes aren't going to do their job as efficiently. So these are just a few things to look for in a client's history. Now, with the liver and the gallbladder, we have our biliary system, which is just a series of ducts within the liver, the gallbladder, and the pancreas that then dump into the small intestine. And bile is made in the liver, and it's concentrated in the gallbladder. So one thing I want to just make sure I make very clear is that the bile composition of the liver is very different from the bile composition of the gallbladder. So when people have had their gallbladder removed, you can't just assume and say, oh, it's fine, their liver still makes bile. Yes, that's true, but the problem is that part of the role of the gallbladder is to concentrate our bile and really increase those bile salts. And it's those bile salts that then help with that emulsification of fat, to then help the digestive enzymes be able to access that fat to really properly break it down. So just be aware that the composition is very different between the liver and the gallbladder bile. But it's made of cholesterol, bilirubin, water, bile salts, and phospholipids. In fact, most of our cholesterol is actually eliminated through the bile. So when people have really high cholesterol, or if they have various conditions that cause elevated cholesterol, like if they're overweight or if they have a really high saturated fat diet, those are going to be elements that are going to oversecrete that cholesterol into the bile and can lead to cholesterol supersaturation of the bile, and that can lead to things like gallstones, because that is where we mostly secrete our cholesterol, is through the bile. Of course, it emulsifies fats, but it can protect against intestinal infections. It plays a role in the immune system, delivers vitamin D from the liver to the intestine, but also bile acids are signaling molecules. So they will bind to various receptors in the body, and they will help to regulate lipid, glucose, and energy metabolism. So bile acids and bile go way beyond just digesting fats. They play a role in detoxification. They play a role in heart health. They play a role in vitamin D in our immune system and in signaling. So we have to make sure that we're supporting and assessing for proper bile flow and function. And then again, if when we eat fat or a meal, that's when that CCK gets released. And that's what stimulates the gallbladder to contract to release bile, and then for the pancreas to release its enzymes. So a few pearls. Gallbladder emptying starts as the stomach begins to empty. So think about how gallbladder emptying and stomach motility are intimately connected. So what happens if somebody's stressed and they have really, either fast, like if it's more of an acute stressor, it tends to speed up stomach emptying, versus if it's more chronic, it tends to slow it down. Or if they have diabetes and they have gastroparesis, how is that going to affect their gallbladder function? It's going to interfere with that signaling. Also, too, the response of the gallbladder to CCK, to tell it to contract, is calcium dependent. So what if people are on calcium channel blockers like Norvasc, which is for hypertension? That's going to decrease the gallbladder's response by 80%. So somebody who has calcium channel blockers, or they, have any issues with their calcium balance, might interfere with how well the gallbladder will contract. And then also, too, think about somebody who's overly stressed, because gallbladder contraction is basically mediated, again, by our friend, the vagus nerve. So, but in a parasympathetic state. So we have to make sure that people aren't eating in a stressed, fight or flight mode. And then these are just some potential signs that you guys can refer back to. Now, this is just a chart that you don't have to memorize at all, but I want you to have it as a reference, because it just has a list of a few, not all, of the hormones in our digestive tract, where they're produced, what they're stimulated by, and also their action. So that way you can have it as a nice little summary, so you can see what stimulates what and what they do. Now, last little section, and then we're done here. But the large intestine, again, 3 primary functions: absorbing water and electrolytes, and then also making sure that we're having good bowel movements. Now, by the time it gets to the large intestine, again, most of those nutrients and about 90% of the water have already been absorbed. So really, it's about absorbing any remaining water, further solidifying the stool, emptying it into the rectum, and then contracting to make sure that we're having a good, complete bowel movement. But this is also where vitamin K and B vitamins are produced by our microbes. So things like folate, vitamin K2, vitamin B1, these are all different vitamins that our bacteria can make. And there's some theory around, if we're deficient or if we have low intake of certain vitamins, that our microbiome kind of compensates and may increase their production of those various vitamins in order to decrease any sort of vitamin disparity. Oh, and here we go, ascending, descending, and sigmoid colons. Now, the ileocecal valve, as I mentioned before, is that little gateway between the colon and the small intestine, helps to keep that backflow from happening. But again, here's a little star, because in the next webinar in November, when you go through the next Foundations series, you're going to talk a lot more about short chain fatty acids and butyrate. So butyrate is super important. It's our main energy for our colon cells. And it plays a huge role in brain health and BDNF and maintaining the epithelial barrier and the mucus barrier. So you're going to go a lot more into that with Kara in November with the next webinar. But the other thing I want to mention is just transit time. So how long does it take for someone to eat food and then see it come out in their stool? Because what I've noticed with my clients and when I've talked with other providers, what can happen is someone might be having a bowel movement every day, but they aren't having good transit time. So have them eat like 1 to 2 tablespoons of sesame seeds, and time when they eat the sesame seeds and then when they see it come out in the stool. If it's less than 12 hours, then they're not absorbing their nutrients and they could potentially have some malabsorption. They have too fast of a transit time. If it's between 12 and 24 hours, that's really ideal. If it's over 24 hours, then that means that the stool or waste is basically sitting in the colon for too long and you're reabsorbing a lot of those toxins. So that's something, it's a really easy way to just assess somebody's transit time and not just rely on, are they having a bowel movement every day? Because there can be a lot of variation in the quality and the consistency and the amount of those bowel movements that can vary between people. So I've had some people where it's been a few days before they see the sesame seeds come out. And so then you need to work on their motility. But the other thing too is, and again, there's a star, you're going to be hearing more about this too when we discuss like more of the hormonal aspect of things, but things like estrogen can be uncoupled. When our body has decided to package up estrogen for elimination and detoxification, as it's going through the intestines, there's this enzyme that microbes will make called beta-glucuronidase that is measured in various stool tests. We measure it on a BiomeFX with the estrobolome. But basically it snips that estrogen nicely packaged up and it opens up that estrogen to then be recirculated back into the system. So you can get various hormonal imbalances if you have elevated beta-glucuronidase and elevated estrobolome in your large intestine. That's going to interfere with estrogen detoxification. So again, some just potential signs of inadequate function, things like diarrhea, constipation, if they're on certain medications, if they have certain things like Ehlers-Danlos, which can affect the function of the ileocecal valve, as well as other connective tissue disorders. That's going to affect all the valves. So your ileocecal valve, your lower esophageal sphincter, those can all play a big role as well. So just in summary, our intestinal and digestive anatomy is really complex. And if we have any disruption in that anatomy or in any of the signaling, that can lead into digestive dysfunction, because there's a hierarchy. There's a hierarchy of flow that depends on higher level signals. And over the next 3 webinars for this series, you're going to further see those dots connected between these foundational concepts as well as how to apply it with your patients. And next time you're going to do a deep dive into the microbiome in November, and you'll expand more on some of the concepts that we discussed today. So I think we've got maybe, I don't know, a couple minutes for Q&A, but I'm happy to stay on for maybe 5 or so more minutes and answer some of these questions if you'd like. Let's see. Angelica, is that okay, or do you need to hop off?
Oh, sorry, I had to clear my throat. Absolutely. We can go over questions. We had lots of great questions in the chat. We also have the Q&A box, but more in the chat, just so you know. So whatever you'd like to do.
Let's see. Let's see about when people have had their colon removed due to disease and what the structure and function looks like for them. Yeah. So Shelley, yes, you're talking about like short bowel syndrome, or if people have had like IBD and they have to have parts of their colon removed, or even some of their small intestine. Yeah, so that is going to definitely, that's like a whole webinar in and of itself, but that will affect the various digestive signals. It affects, actually there's your stomach, when you first have any sort of intestinal removal, definitely if you have any small intestinal removal, you get an overabundance of gastric secretions. So some of the initial issues that people have is actually this overabundance of gastric secretions and gastric juice, which then affects the enzyme output, because again, a lot of those enzymes have to do more, they do better in like a more alkaline pH. So if you have a real acidic pH, you're not going to get proper activation of those enzymes. So yeah, bile flow also changes. There's also more of a potential for like fluid imbalances because of, again, how the colon is so involved in that, water absorption and forming of stool. So yeah, there's definitely a huge role that any removal of, depending on how much of the colon and what parts that can really affect digestive action. Is there a connection between gut microbiome and high folate levels in the blood? Yes, SIBO, actually. So if you have someone who has high folate and they are not supplementing with B vitamins and they're not supplementing with folate, then definitely think about SIBO, because it will cause more folate to be produced, and that will happen. Now, you do want to, with B vitamins, there can be some serum. So definitely, look at red blood cell folate, but yeah, that would be something I'd think about, but definitely over, LIBO, SIBO. And Robert, you're going to be getting, I think, Angelica, y'all are going to be sending out the slides, right, after the webinar. So you'll get a copy of all these.
Absolutely. If
yeah.
you registered for the event, you receive the replay and slides.
Yes. So you all will be getting these charts, and you'll be getting these slides that you can have as a reference for you. Let's see, were there any? Let's see, Rebecca, can you explain coffee and timing of eating and leaky gut? I'm not sure what you mean by coffee and timing of eating. Oh, are you saying like when to drink coffee around the timing of eating and then how that correlates with leaky gut? I guess I'm not really clear on your question. So if you're still on, feel free to type into the Q&A box so I can better answer your question, or send it to the events email, and which I think is just events at, what is your email? What's the events email? Because then I can answer some of these offline if needed.
Everybody could send more questions to my own email address, and I'll drop it in the chat. So if you have any you don't get to, then they can follow up there.
Yeah.
And we'll see if we can answer those.
Yeah. because I'm not sure what exactly. Oh, yes, how coffee affects parasympathetic. So you're asking like, because coffee helps or starts to activate things like cortisol, like that kind of like the caffeine piece with proper digestion. Yeah, so actually coffee stimulates gastrin. So this is why people who drink coffee, sometimes it doesn't sit well, or they might get nauseated, or they could feel kind of more of that acidic well, it also releases the lower esophageal sphincter, but yeah, it definitely is going to affect gastrin release and can affect the stomach acidity levels. I guess I'm still not quite clear on but yeah, if you have caffeine, I mean, usually if I have a patient who is like really stressed and they already have really high cortisol, I tell them to not do caffeine because that's only going to perpetuate the whole like cortisol response. And so, yeah, it's interesting how sometimes people feel so much better just taking the caffeine out because they're in a sympathetic state. And so helping to calm things down and switch to like a decaf or like a coffee alternative can really help to reset. Because again, if someone's in that is in that sympathetic state, over time, you're going to chronically suppress and you're going to chronically interfere with that vagal signaling for proper digestion. So I think that's what you're asking. And I absolutely agree that if someone's in that, like if we're having to work a lot on sympathetic issues, I take coffee out and I take the caffeine away because that's only going to exacerbate it.
We had a few in the Q&A box, Alicia. I don't know if you saw those.
Yeah, I think I answered those.
Oh, you did? Okay.
Do you have any sense of what percent of guts go dysbiotic after colonoscopy, like oral? Yes. So colonoscopy, there's actually been research on this. Colonoscopies, they cause an imbalance in the microbiome. I've seen anywhere between 2 weeks up to like 2 months, depending on the study that you're reading after colonoscopy due to the prep. And there's some research that shows that the microbiome goes back to normal after that 2 week to 2 month period. But then there's some papers that also show that once you've done a colonoscopy prep, there are some families and there are some microbes that don't go back, that actually are altered after colonoscopy. So there's some information out there about that. And some of the imbalances that can happen. And it just, you just kind of have to look at the different papers, but it's interesting how some of the, like lactobacillus I know is affected. There's a few others that are affected. So what I recommend usually if someone's had a colonoscopy is that you really do want to work on post-colonoscopy, you want to really work on getting back to that eubiosis and work on some of those dysbiotic factors too. Let's see, suggested test for an oral microbiome. I think there's a test called Bristle that does that, I think that does that. And will you be using products? Yes. So I think it's our last Foundations webinar that we'll talk more about products and protocols. Mostly we wanted to make this as just like a learning opportunity for you guys and not a ton of product focus, just giving you guys some really good science and information that you can take away with your patients. But we'll kind of tie it all together in January and with the final 4th webinar. We'll bring in a lot of those like, okay, what do you do about it? Kind of things. So, well, digestive enzymes. So I'll probably answer like maybe 2 more questions just for time. Will digestive enzymes and betaine HCl impact the natural ability for the body to perform without them? How long is too long of use for either of these? Will berberine negatively impact the use of spore-based probiotics? So digestive enzymes in general don't really, although you can recommend that people take their digestive enzymes in the middle of a meal or at the end of a meal so that your body has a chance to naturally try to do it on its own first, and then you're just kind of bringing it in and you're supporting it kind of at the middle, the end of a meal. Betaine HCl, you just have to be kind of careful with it because if you do the betaine HCl challenge where you're adding it in and then you're gradually titrating up till they feel that warm sensation, some people don't ever feel the warm sensation, and then you have people taking like all this betaine HCl, and that actually, and I've seen this happen with other practitioners, providers, and whatnot, where it actually gives people an ulcer because they were taking way too much betaine HCl with each meal. So just be really careful with it. The other thing is if you over acidify, then again, go back to our digestive enzymes have to have a proper pH to work. So if you're giving all this like 6 caps of betaine HCl with a meal, that's a lot of HCl. So I usually like to use things like bitters, digestive bitters, things that are more natural, having them do some apple cider vinegar and water, just more as a supportive option. And also encouraging your body's natural secretions of those versus trying to layer in with a bunch of betaine HCl. So I usually am real cautious with that use. And then as far as berberine, so berberine, there's actually, it won't, I would probably not take it at the same time as the spores, like in like the same meal or the same swallow. But you could take it like in the same day. And berberine has some really good, it's actually got some pretty good data on it around, it hasn't been studied a lot in like IBS patients populations, and it hasn't been studied long term, but short term at like moderate dosages is actually pretty useful and pretty impactful. And berberine has had a lot of really good data on like blood sugar and metabolic support. So, but yeah, you just want to be really careful if you're doing really high dosages of berberine, that could start to negatively affect your microbiome.